So hi everyone. Welcome to this sort of SCSE Exploration Club case study for this week. We're excited that new users or users that we've had in the previous sessions are joining today. Sort of the main focus of the session today will really try to utilize our Talk to Data system, so our new version 5 of the Talk to Data, to really explore several different single cell case studies focusing on astrocytes and alpha-synuclein in the context of Parkinson's disease. So we'll try to sort of leverage these multi-studies that we have relating to single cell RNA-seq within the human database to really answer sort of this focus question. And our main goal is really to try to understand how astrocytes respond to alpha-synuclein across different brain regions, and specifically whether the expression of this particular sort of gene, so the gene SNCA, which is the encode of the alpha-synuclein protein, is sort of lowly expressed within astrocytes, representing whether it's a protective or a dysfunctional state within this disease landscape or neurodegeneration as a whole. So that will be sort of the focus. We'll utilize a few different features from the Talk to Data version 5 system to really look at transcriptional differences, correlation analysis, and really just mapping out the expression of this alpha-synuclein, and if we do in effect see low expression from what we see from previous studies, and different expression across different regions of the brain. So that will sort of is sort of a nutshell of what the sort of case study will be on today. So just a recap on the previous sort of week. So as you know, the SCSE Club is sort of delivered to you weekly. We try to do a wide range of different case studies to really sort of help and support different research questions that users using the system currently are able to really utilize and really understand how they can move their research forward from these sort of examples. So the episode from last week really tried to focus on our other module, Spatial X, which is more of a spatial transcriptomics sort of platform and multi-image integration platform. And what we saw from the previous results, just as a recap, is that we were able to identify neurons expressing CLC17A7, oligodendrocytes that also had a specific profile expression of MOG, which identified very specific spatially localized sort of markers using different models like the cell type prediction. We also sort of covered region segmentation, which is another feature in Spatial X, which really allowed to define 5 different spatial domains, and particularly a specific segment that had sort of a neuron-enriched marker, which is the CLC17A7. And other sort of results that we were also able to gather, if you have an interest in Spatial X and are looking into analyzing spatial imaging data, is really looking at sort of Alzheimer's disease neurons and sort of specific transcriptomic or transcriptions factors that sort of are downregulated and have an increased sort of role in either the co-localization of certain cell types, so in this case, endothelial cells and parasites. So these are things that we covered last week. So if you do have an interest in sort of Spatial X or just spatial transcriptomics analysis and you're new to that field, you can look at our previous videos on our YouTube channel as well. Okay, so just before I go into the actual live demo on how to actually perform these different analyses and what research questions or objectives we'll try to answer, I do want to give some sort of some background on really what alpha-synuclein is, or what is its central role within Parkinson's disease, so you sort of get an idea. So Parkinson's disease, if you're not currently working within sort of the neurodegenerative space, is classically defined as the accumulation of alpha-synuclein aggregates, so really focusing on this particular protein, which is the product of the CNCA gene, into Lewy bodies. And sort of we see this particular sort of protein at a loss of dopaminergic neurons in the substantia nigra. These aggregates are not only a hallmark of this disease, but also drive pathology as well, by impairing either the cellular proteases or the mitochondrial function or somatic transmission. And really what the main attention should be is focusing really on how alpha-synuclein's role sort of affects or is sort of involved within neurons, and how it's sort of been established to extend also to glial cells. So something that we'll really try to look at is not only its role within sort of Parkinson's disease or neurodegeneration progression, but also how the pathology extends to other cell types that might not have been the focus on other sort of literature, which is glial cells or more specifically astrocytes. So we're going to sort of look at how these sort of expression of this protein is altered within astrocyte population specifically. And can we relate that to stress response or disease progression in sort of Parkinson's disease as a whole? Okay, so another, I guess, side of it as well is really understanding how astrocytes transition from either being protective, so neuroprotective within sort of the that space, or sort of leading to neuroinflammation. So these specific cell types are quite interesting in the way that they do transition in different sort of functional roles, and it really depends on the disease context as well as what cell types are sort of involved within this transition. So astrocytes as a cell type really play vital roles when it comes to maintaining neuronal health by clearing, in this case, alpha-synuclein. And this sort of leads to the regulation of glutamine metabolism and also producing antioxidants and supporting mitochondrial function. In early Parkinson's disease, we normally see astrocytes that may try to help protect neurons by uptaking or degrading this alpha-synuclein protein. However, there's also literature suggesting or sort of showing that there's a persistent sort of alpha-synuclein exposure. And this really shows it by astrocytes undergoing a shift within this environment. They become more reactive. They overwhelm sort of the intercellular aggregates of this sort of protein. And this, again, leads to this sort of mitochondrial damage, inflammation, and other releases of pro-inflammatory cytokines as well, which can be IL-6, IL-1, or TNF alpha. And they might switch between these different phenotypes, whether they're protecting or in the sort of late stages, they're sort of showing this neuroinflammation role. So this is what we'll try to really look at, but not only in a single study, but really trying to understand if we're able to get more answers from the talk to data system and more so across multiple data sets that we can really sort of isolate these astrocytes from. Okay. And one last thing that I wanted to cover is really understanding the expression of this particular gene encoding from the sort of alpha-synuclein protein, how it's expressed in astrocytes, and particularly in Parkinson's disease, as opposed to what you would see in healthy individuals. So despite sort of seeing that evidence that alpha-synuclein aggregates in astrocytes show a low or absence of this CNCA expression, we do see that this is really dependent on what brain region the sort of study was analyzing. So really our focus today as well in one of those aims is really trying to understand how we're able to look at pathway enrichment, but particularly in astrocytes either expressing low SCNCA in specific brain regions where we see that low expression, or brain regions where we see a high enrichment of this SNCA. So we'll try to sort of run differential gene expression from these cells from these different brain regions that I'll sort of cover later on in the session. Okay. So one thing that I want to mention is really how are you able to do this from the talk to data system? So can we actually integrate multiple studies? Can we isolate very specific cell types with this sort of expression profile of this gene encoding protein? Yes, we can. So one of the ways that you can do that is from the talk to data version 5, which will require some filtering. So it will require you to kind of know what sort of gene you're really focusing on, what brain regions are you focusing on, and really trying to understand just like how is this gene sort of expressed not only in astrocytes, but across all different cell types. So can we really sort of identify the role of this gene first, and then can we go deeper into isolating astrocytes within different brain regions? So that will be sort of, again, the main focus is really mapping the expression of this protein, sort of alpha-synuclein gene encoding sort of gene, which is a CNCA, across different sort of single cell RNA-seq data sets. So you'll see here from this sort of example, we have currently in the database from my filtering, just looking at astrocytes, we have 14 different studies that have isolated or have analyzed astrocytes, but probably did not really focus on astrocytes within the actual study. But they have sort of identified these cell types in these different data sets. So these are the ones that we'll try to sort of integrate into answering some of our questions. So the agenda for sort of this session will focus on three different areas. One, which is how can we characterize SNCA expression across all cell types and all brain region tissues in Parkinson's disease data set. So can we actually identify very specific differences, even just looking at different cell types or different brain regions? The second part will be, okay, from there, can we isolate or identify astrocytic transcriptional program differences, which are associated to either low or high SNCA expression, and can we isolate those specific brain regions from those studies only? The third one we'll also try to really cover, if we have some time, is really exploring correlations between NC and SNCA with other genes, and can these other genes give us some indication of their role in inflammation, stress response, or also just sort of protection as well. So that will sort of be a last thing, but if we don't have enough time, we can definitely do a follow-up session or one-to-one if you are interested in this area as well. Okay, so for the talk to data version 5, which I'm showing an example here, the main research question that we'll try to really answer again is SNCA expression in astrocytes and how they vary across different brain regions, disease states in Parkinson's disease, and can we distinguish very specific transcriptional features within low or high expression of this protein encoding factor that our sort of astrocytes are expressing within this environment. So that will be sort of the focus again. So now we'll go into the live demo, which the first part will be really how can we define this expression distributed across different major brain cell types or different sort of tissue regions as well. So in order to do that, we'll go into sort of our biotrain ecosystem here. We'll find that our talk to data module is here on the right-hand side. So we can directly access the talk to data system, which it'll sort of show you that you can now access the new version 5. So we can continue with that version 5. And the next step will be to really input our specific gene factor or a protein encoding gene into the system. We can search for the SCA particular marker, and we'll try to sort of add filters to really only focus on Parkinson's disease. So normally when you add a specific gene search, it's going to run it across the entire system. So it'll show you the sort of results based on a sort of ontology tree structure, where you're able to see the expression of the gene just across all cell types in the entire database. It's not disease specific, so it's just showing you where is this gene really expressed across different diseases. If you want to then really focus and sort of make it very specific to a disease area, you'll need to add very specific filters into the ontology tree. And that really focuses on specific standardized ontologies that are mapped across these different studies. And it's just a way that we normalize all of the studies in the system so you're able to really run this cross-study analysis focus. So on here, what I'll do is add our Parkinson's disease focus, as well as other samples that might be relating to Parkinsonian disorder. So you'll see that there's different standardized labels depending on the actual ontologies that are coming from the studies, and then how do we normalize and map them into sort of a standardized ontology system. So when we apply this search, it'll automatically add that filter for only those samples within those disease areas. So from here, what we can do is then make sure that we have the gene expression of our particular protein encoding gene. We can then apply that search, which will then sort of focus within these different cell types. So what we can see just by looking at Parkinson's disease across these different studies that are expressing this particular gene, we can see that there's specific cell types that are sort of shown more heavily. So what is sort of the results that we're able to see just by looking at the different cell types or different tissues? So what we can see is that neurons show a highest expression, which is this sort of level not here. And we can actually also change this if we're adding other filters like cell types. If we want to focus on only certain populations, we can view those selected nodes only, or those cells that are coming from those selected nodes or those filters. So that's also something to keep in mind. So when we apply that search, what we're able to see, looking at the gene expression of this particular factor within these samples, we can still see that it's mainly sort of focused, and we can also sort of change the average rank as well. So if you're mainly focusing on specific cells that are expressing only sort of a certain level of enrichment or an average rank expression level, you are able to also narrow down that filter. So we are able to see just by looking at the scale that mainly neurons show this highest SC and CA expression, particularly sort of excitatory subtypes of these sort of cell types. We can also see that astrocytes, which are one of our main focuses of today, show a lower expression, and it's coming from 14 different studies, particularly only isolating these astrocytes from these studies. And this is sort of showing this absent transcription across different sort of regions. So if we really want to see, okay, how is this consistent in different regions of the brain? Can we actually look at these differences? You are able to do that. So if we want to go into that specific area, we can go into this specific filter here and actually look at the anatomy of different brain regions. So looking at the expression of this particular marker or transcription factor within these samples, we can see that it's specific to very specific brain regions. So in one end, we do see that it's also enriched in sort of blood samples. So if there is an interest in really exploring sort of different blood samples compared to different brain regions or different tissues, that would be sort of one focus. We can also see that just within the brain region, we are able to see that the main expression is really within sort of the midbrain. So we can see that mainly the substantia nigra is showing this sort of expression of these SC and CA. As opposed to other regions that might be more like cortex or primary motor cortex or other sort of areas in the forebrain. So this is where you're able to really look at where can we see this expression within the nervous system. So if you're particularly only focusing on different brain regions, this is where you are able to look at that expression. You can also change the average rank to really look at where that enrichment is mainly concentrating on. In terms of how you're able to change the scale as well. So if you're mainly focusing on looking at not only the relative. So when you adjust the average rank scale, what you're trying to do is configure how you're looking at the expression of SNCA within the entire database. So in the absolute mode, what you would be viewing is the node scale to the entire database and data range. So it's not particularly looking at the gene's minimum or maximum range within those filters. So if you're interested in a more sort of over representation of the entire database where it's scaled to that sort of unit, you can do an absolute scale. Or if you're doing it based on the minimum maximum within those filters, you can make it relative to those sort of filters that we've added in the database. So this is sort of an example, just looking at sort of how SNCA is really enriched in neurons. We can see that astrocytes or other microglia show minimal transcription expression of this encoding protein sort of gene target. And we can also sort of get an idea really how this supports how astrocytes respond within, or how they produce sort of this α-Synuclein protein. And can we look at this across only astrocytes? So if you want to add that second layer where we're only sort of isolating astrocytes within this specific analysis, we can add these cell types here and apply that specific filter. And that will then show you the role of these astrocytes, and particularly looking at this sort of alpha-synuclein protein and how it's either aggregated or it's either sort of having other functional effects within sort of Parkinson's disease. So just by filtering, we can see that if we're only focusing on the role of this particular gene within astrocytes within these samples, we can see very clear differences. So we see an enrichment within the midbrain region, particularly the substantia nigra region, as opposed to what we see in the forebrain or the what we would call the cerebral cortex regions. So we see less enrichment of this particular alpha-synuclein that are specifically sort of expressed by these astrocytes across these studies. So this is a really important, really analysis that we can then move forward. So we've now sort of uncovered across these 14 data sets that we can actually see very specific transcriptional differences that are showing differences in the aggregate of this sort of alpha-synuclein within astrocytes across these different brain regions. So if we want to look at further transcriptional programming or reprogramming and enrichment of specific pathways within this reprogramming sort of function, we can then perform differential gene expression from these results or these cells in these data sets specifically. So if we want to do that, the next step would be to then use this data to go into the differential gene expression feature. So the next step after we really uncovered these specific differences in low or absent astrocytes within Parkinson's brain regions and really looking at how this gene is expressed not only in neurons, but also showing this low expression in glial populations, we can then determine which genes and pathways are actually enriched in astrocytes that are showing these differences in low or high expression of this particular encoding protein, and whether these represent very specific neuroprotective or dysfunctional states. So can we actually really look at sort of these processes in the database? So in order to do that, again, we'll go into the differential expression feature. We'll try to sort of add what exact analysis we're doing. So in this case, we'll be looking at, if I go back to the DG history, we'll be looking at astrocytes, transcriptional differences based on PD samples. Or you can sort of add any specific sort of title to the sort of analysis so you're able to go back and know exactly what groups you were trying to compare. From here, we'll need two different groups. So in this case, since I'm interested only in Parkinson's disease, it'll have very specific filters that will be the same in both groups. So one of them will be the actual condition focus. So really looking at only Parkinson's disease in this case. Okay, the next step will be the cell type. So again, we only really want to look at transcriptional programs of these sort of astrocytes within PD. So again, we'll add this astrocyte filter, and this will sort of be the same on both groups just because we are trying to compare Parkinson's disease and only astrocytes, but it will differ on the actual brain region that we want to look at specifically. Okay, so we'll add the Parkinson's in group 2 and also astrocytes. Okay. And for group 1 in the anatomy, this one can be focused on, so if we want to really focus on two different groups, one of the ones that we saw from the previous results, so when we looked at how can we map this from the ontology or creating different bubble heat maps to see the tissue differences, one of the main groups was that we saw high enrichment of this sort of alpha-synuclein within the midbrain region of the astrocyte, so really the substantia nigra region. So in this case, what we can do is add midbrain in group 1 and also look at substantia nigra as well if we want to add that sort of further closer look or that granularity. So in this first group, this will be sort of midbrain region where it had sort of a high SNCA environment. So we can add that in the title. Okay, and the next group will be really looking at low SNCA sort of environment. So really the prefrontal cortex or the neocortex. So in this case, it'll be the cortex area or the cortical region, which had a low SCNCA. Okay, so in the anatomy, I can add here the sort of prefrontal cortex or the neocortex. Okay. And this is where it's going to show us how many cells do we have from these different studies relating to these different regions and this expression of this particular gene within these astrocytes only. So it's just going to show you how many cells we're actually training from these different groups or we're actually collecting from the studies. And from there, we can start running the analysis. So it'll sort of show you the system when it's processing. And once it's done, it'll show you sort of this success rate as well. So what can we see from here? So normally when you run the differential gene expression analysis, you're able to sort of get this sort of full list of differentially expressed genes with different log fold changes across both directions. So you can actually identify genes that are upregulated within these two different groups or downregulated within these two different groups. So you are able to focus either if you're looking at upregulation or you want to focus on markers that would be downregulated. So you're able to know what treatments you want to focus on or what are your targets that you can actually use. So that's something that you can actually filter in the list. So you're able to define those markers. You can download the entire data as well, or you can actually use the system to view the full report. So in the full report, what you're able to see is the list, and you're able to look at a volcano plot of those specific markers that are either upregulated or downregulated within the study, and which ones are actually important within the results. So something that we can do here is actually looking at the top 10 genes. We can also select if we want to look at it across different direction. So if we're interested in upregulated genes, we can press OK and we can add top 10. And we can then apply that as well within either the midbrain astrocyte region or the cortical astrocyte region that had these differences in the expression of this protein encoding factor. So what can we see in just the heat map view? And I'll sort of show you from the slides because it's easier to actually view as well. So when you're in the system, what you can see is that there's actually very specific targets that are either upregulated. You can look at both directions as well. So if you want to see genes that are going in both directions, you can apply that search as well to see if there's any differences within the groups. So something that we can see just by looking at the heat map is that there is a difference within this particular gene, the AAK1 gene, which is highly enriched within the low environment of SCNCA within the cortical region, and particularly astrocytes that are sort of located or aggregated within the cortical region in the brain. We can also see that there's other less expression of other genes within the midbrain astrocyte region. So if there's an interest in really looking at just all the genes and what can we actually see is that astrocytes in midbrain, so high and SCNCA environment, show a clear transcriptional reprogramming, including upregulation of stress or inflammatory regulators. So we can see that by looking at the volcano plot as well. And there's a downregulation of homostatic genes as well. So now the next step, really looking at or moving these genes further, is really looking at what pathways are actually enriched within these different groups that show this sort of transcriptional reprogramming, and can we connect that to functional pathways that actually relate to these differences. So in that case, you do have that option within the Talk to Data system. So really cells coming from these 14 data sets to really run pathway enrichment from those targets that are either upregulated or downregulated. So that's something that you can select as well. If there's specific markers that you're focusing on, you can focus on those genes, or you can just look at all the genes analysis. So something that we'll want to look at is pathways relating to the stress response, so reactome. Specific database, and also biological processes to look at either mitochondrial changes or lysosome changes that might be leading to this aggregate of this alpha-synuclein protein and the stress response in these brain regions. So something that you can do when you're looking at these results, you'll have the two different pathway databases as well as the entire list of these pathways. So looking at either biological or reactome specific pathways, and then you'll see how these are enriched within different directions. So are these actually upregulated within the samples or downregulated? So something that I can show you just by looking at the results of the pathways is that we are able to see that there is sort of a Parkinson's disease related astrocyte stress across, if we're looking at specific astrocytes that are expressing either high alpha-synuclein or low alpha-synuclein, and really what brain region is that really in. So we can see that within the midbrain, so the high SCNCA, compared to the cortical, which has a low astrocyte expression of this protein, is associated with proteostatic stress and sort of transitional control. So what we're able to really look at is within the low expression, particularly, we can see that there are pathways relating to mediated decay. We can see other pathways related to DNA repair as well, RNA methylation, or other sort of pathways that might be relating or consistent with oxidative stress or impaired homeostatic function as well. So that's something that you're able to also run from all of these different astrocytes across these different data sets. And really, again, now that we're sort of come to the end of these specific functions that we covered, what is sort of the summary that we were able to gather from these results? So something that we were able to see just by really mapping out different sort of expression of the sort of alpha-synuclein encoding protein factor gene is that we were able to see that SNCA is expressed in neuron-enriched regions and that are high in the midbrain region, so particularly the substantia nigra region, as opposed to astrocytes that normally show a consistently low expression across sort of specific cortical regions in the brain, supporting that there is a non-transcriptional sort of alpha-synuclein uptake within these specific cortical regions or sort of motor cortical regions in the brain. Within these sort of Parkinson's disease patients. The second part, which was really once we isolated only astrocytes and we wanted to look at further the transcriptional differences, so really utilizing the differential gene expression feature, we saw that the midbrain astrocytes that had this high enrichment of this encoding factor showed an upregulation of stress-related genes. So for example, NEAT1 or SOX2, which was shown from the volcano plot here. So we can see very specifically that in the high environment, there's very specific sort of other markers that are encoding this specific sort of function. Whereas when it came to the low region, so the cortical region, we saw downregulation of hemostatic regulators. So there is sort of a reactive transcriptional shift. So we saw this GGAT1 factor. We saw other sort of genes as well within this sort of downregulation in the midbrain. So now if we go into the other last sort of summary is really the pathway enrichment analysis. So how can we take this, this finding, this reactive transcriptional shift, and can we actually connect that to functional pathways from the database? So we were able to run pathway enrichment analysis, which then looked at or really uncovered activation of translation pathways, nutrient stress, or proteases pathways within the midbrain astrocytes, which give us more indication of this suppression or this sort of alpha-synuclein induced cellular stress. So these are sort of key analysis or key results that we were able to gather just by looking at the talk2 data system, which does give us a lot of data that we can actually utilize for grants, for validating our own analysis that we can see from our own study. But this is being trained on several different data sets that are normalized and that are mapped on very specific ontology systems. So again, now that we came to the end, my colleague will be sort of circulating a poll if that hasn't been done already. So the poll is quite important because it gives us an idea if this case study was really useful to you, or if there's other case studies or other topics that also might be relevant to your analysis that would be useful to really explore within the database or other modules within the database. So please, if you're able to just give us some insights in what you thought about the case study or what would you like to see in the next case studies, that would be great so that we can do that for you. Okay. If there are any questions from anyone who's joined us today, you can either send it through the chat if you want it to be answered today, or what you can do is also reach out to the support email and we can answer those questions as a one-to-one session. So Kisha, I can see that you can't view the whole screen. So that might have been from the previous run. Sorry if it wasn't clear on your end. If there is something that you want us to show, or if you want to see the slide so we're able to show that result, that we can send that to you as well. That's why we usually like to have it in slides so you're able to see the actual results or the specific figures in the system, because they are quite small if you're looking at many genes. Okay. So if there's any other questions, just let me know. So I think there's no other questions in the Q&A. So I'll let just a few seconds just for anyone else who's joining to answer that poll. And again, if you have another interest in really running a one-to-one session to go into these features a bit slower or just with more detail, you can reach out and we can go through your case study or your disease area within the talk2 data system. Okay. Okay, so thank you everyone for joining. So I think we gave enough time for the poll. So I look forward to seeing you in the next session. We haven't yet defined a specific topic, but we'll circulate that so you're able to know next week's Exploration Club topic. So thank you everyone for joining today. And I hope this was a useful case study for your own analysis or just something that might have been of interest to you in exploring. Thank you.